This video is about the stages of mitosis, and those stages are prophase, metaphase, anaphase, and telophase. Mitosis is a form of cellular division, and all living things engage in cellular division. And cellular division is used for reproduction, growth, and repair. There are three forms of cellular division, and they are binary fission, mitosis, and meiosis. Mitosis is eukaryotic cellular division. It's a form of cellular division that occurs only in eukaryotes. And mitosis involves DNA replication, the copying of the DNA, division of the nucleus, and cytokinesis, when the cell splits in half. To understand the steps or the stages of mitosis, we have to understand what chromosomes are. All living things have DNA, deoxyribonucleic acid. And DNA stores genetic information. And DNA forms chromosomes. So the image on the bottom, there's some DNA. And on the right, we have a chromosome. And that chromosome is made out of DNA. It's a whole bunch of DNA, and it forms a structure that looks just like that. Chromosomes can exist in either an unreplicated state or a replicated state. And replicated means copied, and unreplicated means not copied. On the right, in the green outline, we have an unreplicated chromosome. It's one copy of a chromosome. But next to it, in the red box, we have a replicated chromosome. On the left side, in the red box, we have one chromosome, and it's going like this. So don't think of this as an X. Don't think of it like, let's find a different color. Don't think of it like, we have one chromosome here and another here. It's not like that. Follow the yellow. There's one chromosome here, and then there's a second over here. So on the left, there's a chromosome, and on the right, attached to it is a copy of that chromosome. We don't call these chromosomes at this point. We say that this is one chromosome in the red box composed of or made out of sister chromatids. So let's look at our bottom bullet point. In a replicated chromosome, we call the two copies of the chromosome sister chromatids. Now eventually they'll be pulled apart and we'll call them separate chromosomes. But at this point, it's one chromosome composed of sister chromatids. And the sister chromatids are identical copies of each other. They're exactly the same. During mitosis, the sister chromatids will be separated. At that point, we will call them separate chromosomes. But until then, we say that they're part of a single chromosome. They're sister chromatids making up a single chromosome. Chromosomes can be either condensed or uncondensed. Condensed means tightly packed. So obviously uncondensed means not tightly packed. Uncondensed chromosomes lack a clear shape, whereas condensed chromosomes have a clear shape. On the right, that sort of purple blur, that represents an uncondensed chromosome. And think of the image on the right as the same chromosome, but now condensed. You can see it now has a clear shape. During much of the cell cycle, chromosomes are uncondensed. So that sort of purple blur, that's what they usually look like. But during mitosis, chromosomes are condensed. And that image on the right, it's actually much easier to see. So that's become the classic image of a chromosome, even though chromosomes are actually usually in that uncondensed state. But it's during mitosis that chromosomes become condensed, and they assume that shape on the right. Both uncondensed and condensed chromosomes are composed of chromatin. Chromatin is DNA wrapped around proteins. When we speak about chromatin, we're usually referring to the uncondensed state of a chromosome. So when you think of chromatin, think of that purple blur. But when we speak about chromosomes, we're usually speaking about the condensed state of a chromosome. Even though both of these images on the right, they're both made of chromatin. but often the word chromatin will be used to refer to that purple blur. And chromosome, when someone says chromosome, you want to think of that image on the right. The lifespan of a cell is called the cell cycle. And the stages of the cell cycle are G1, S, G2, and M. During G1, the cell is growing. During the S phase, the DNA is replicated or synthesized. Synthesized means made. So it's another way to refer to the same process that we're going to call DNA replication, where the DNA is copied. G2 is when the cell prepares for cellular division. In the M phase, that's when the cell actually divides. It's going to go through mitosis and then cytokinesis. At the beginning of the cell cycle, the chromosomes are unreplicated. They haven't been copied yet. DNA replication or synthesis hasn't occurred yet. If we look at our cell, we can see it has a nucleus. This purple oval is supposed to represent the nucleus. So we know this is a eukaryotic cell. It also has other organelles that we associate with eukaryotic cells. These guys here, and we have the rough ER over here. It's hard to see my highlighter. I was trying to point to that. These are the unreplicated chromosomes. These guys here. So notice it's just one line. It doesn't have those two lines kind of attached to each other. So those are unreplicated chromosomes. Now, one thing about this image, though they are illustrated in a condensed state in this image, chromosomes are in an uncondensed state throughout interphase. So I've illustrated them in a condensed state here, just so I could let you see that they're in an unreplicated state. But they're actually not condensed here. They're in a loose, shapeless state. During S phase, the uncondensed chromosomes are replicated or synthesized. So it's during S phase that the chromosomes make copies of each other. They're going to become sister chromatids. Although, again, because this is still interphase, they're in an uncondensed state. That's why they're illustrated as that sort of purple fuzz or purple blur here. That's the chromatin. That's the uncondensed chromosomes. When the cell enters M phase, the chromosomes condense. And because they were replicated in S phase, they're now in a replicated state. So here we are, we have our replicated and condensed chromosomes. When we see this, we can know that this cell is about to undergo mitosis. This image here of condensed replicated chromosomes is a sign that this cell is about to undergo mitosis. To put it all together, at the beginning of the cell cycle, the chromosomes are unreplicated. And throughout interphase, during interphase, the chromosomes are uncondensed. But during M phase, the chromosomes are condensed. And they've also been replicated by that point. So if we look down the bottom left, we see we have our unreplicated chromosomes. And in the image in the middle, that's actually what the chromosomes look like throughout interphase. They're just chromatin. We can't see the chromosomes. And this is actually what the chromosomes look like. They're in this uncondensed state of chromatin, loose chromatin, during S phase when the chromosomes are replicated. But on the right, we have our condensed, replicated chromosomes. And we know when we see this image that this is a cell that's about to undergo mitosis. Mitosis takes place in stages. Those stages are prophase, metaphase, anaphase, and telophase. And PMAT is an acronym that's supposed to help us remember those phases. The phases or stages of mitosis are numbered in the images down the bottom. The first stage is prophase. The second stage is metaphase. The third is anaphase. And the fourth is telophase. You can see one of the most important aspects of mitosis is going to be what's happening with those chromosomes that you can now identify there. You can see the chromosomes in prophase. And then notice they're all lined up in the middle in metaphase, the second phase or stage. They're being pulled apart in the third phase. And then the fourth phase, what you can see there is actually new nuclei developing around the chromosomes. Eukaryotic cells possess organelles called centrioles. And centrioles facilitate mitosis. And to facilitate means to help. So they help cells undergo mitosis. And spindles extend from the centrioles. If we look over here, we have our centrioles. Let me grab a different color. Red. So here are our centrioles, and these are the spindles. So you're going to see these throughout mitosis. And now you know what they are. There's the centrioles with spindles extending from them, and they help the cell undergo mitosis. They help it carry out mitosis. This is an image of a cell in prophase, the first stage of mitosis. During prophase, the chromosomes condense and a nucleus dissolves. During prophase, the pairs of sister chromatids appear. Thinking of this as the stage in which the pairs of sister chromatids appear might help you because you can think, well, PMAT, the first stage is prophase. What happens during prophase? Well, the pairs of sister chromatids appear. So that's just a trick for trying to remember these stages and what's happening during them. But let's look at the image quickly. These are our, you can't see it when I highlight it, but what's going on there with the chromosomes is they're in a replicated, condensed state. And that's because this cell has just entered mitosis. And this here is our nuclear envelope. It's part of the nucleus. And notice it's dissolving. So the nucleus is going away at this point. During metaphase, the chromosomes line up in the middle of the cell. So PMAT. The second stage is metaphase. What's happening? The chromosomes line up in the middle. If you look at the image, the chromosomes are all lined up in the middle. And also note that the nuclear envelope is gone. It's completely dissolved. So that's because, remember, we're building two new nuclei. This is division of the nucleus. That's what mitosis is. So we got rid of the original nucleus, and now we're going to pull these chromosomes apart so we can build new nuclei. It's during anaphase that the sister chromatids are pulled apart. So we're in our third stage of mitosis, anaphase. And that's when the chromosomes are pulled apart, pulled away from each other. In the bottom on the left, we can see two sister chromatids being pulled apart. And we've seen that before when I was talking about chromosomes being pulled apart. Remember, originally that was one chromosome composed of sister chromatids, and now we pulled them apart, and now they're separate chromosomes. And if we look at the image on the right, you can see it's exactly what's happening here. Notice the centrioles with their spindles, that's what makes this happen. And so obviously those centrioles and those spindles, they're actually essential to mitosis. But the point here is that in anaphase, the sister chromatids are pulled apart. They're pulled away from each other. During telophase, new nuclear envelopes develop and the chromosomes decondense. Right, so I've added a D here. D means it's undoing the condensing. So remember, the chromosomes condensed during prophase, and they've been condensed throughout mitosis. But during telophase, they're going to decondense. They're going to revert back to just being loose chromatin. And in telophase, two nuclei develop. If we look over here, we see a new nucleus is developing. And the same thing over here. So this cell, for a moment, this won't be very long, but for a moment, it's going to have two nuclei inside of it. So that's what's happening during telophase. The chromosomes are decondensing, they're reverting back to being loose chromatin, and two nuclei are developing. They're going to be the nuclei of new cells. Mitosis is followed by cytokinesis, and we can see on the right what's happening here is we have our two nuclei that developed during telophase. They're filled with loose chromatin because the chromosomes decondensed, and the cell is dividing in half, and that's cytokinesis. So during cytokinesis, the cytoplasm is divided. During cytokinesis, the parent cell divides into two identical daughter cells. Remember those chromosomes that were composed of two sister chromatids? Remember, we said each was identical to the other. And then we saw they were ripped apart during anaphase. Well, those chromosomes, those sister chromatids, one cell got half of the sister chromatids. The other new cell, the other new daughter cell, got the other half. So these cells have identical chromosomes inside of them. So they're identical daughter cells. So hopefully this image is clearer now. We said mitosis has 4 stages: prophase, metaphase, anaphase, and telophase. We have the acronym PMAT to help us remember it. And number 1 down on the bottom, that's prophase, where the pairs of sister chromatids become visible because the chromosomes are condensing. And also the nuclear envelope is dissolving. And then in the second stage, metaphase, the chromosomes line up in the middle and the nuclear envelope is totally gone. In the third phase, the sister chromatids are pulled apart or away from each other. In the fourth stage, well, I should say the third stage was anaphase, and in the fourth stage, telophase, 2 new nuclei develop, and the chromosomes decondense to become loose chromatin. And on the right, we have cytokinesis. This is when the nuclear envelopes have fully formed, the chromosomes have completely decondensed into loose chromatin, and the cell is finally going to divide, what we call cytokinesis, and become 2 identical daughter cells. The purposes of mitosis are reproduction, growth, and repair. We have an image of a hydra with a bud growing off of it. The cells in the bud are produced through mitosis, and that's a form of asexual reproduction, budding. The plant growing on the right, when multicellular organisms grow, they have to add new cells, and they add new cells through the process of mitosis. And of course, the Band-Aid is intended to represent injury. When tissues are damaged in multicellular organisms, well, only multicellular organisms have tissues, but when their tissues are damaged, the organism has to add new cells to replace the damaged ones, and it uses mitosis to do that. And cells can be damaged through just natural wear and tear as well. In the stomach, the lining of the stomach, the cells that make up the lining of the stomach, are frequently replaced because they're damaged just as in the process of performing their usual functions. So it's just normal wear and tear. And mitosis is used for that purpose as well. Mitosis in the narrow sense, division of the nucleus, is followed by cytokinesis. That's when the cell actually divides in half. Cytokinesis proceeds slightly differently in plants than it does in animals, and it all has to do with the fact that plant cells have cell walls. So plants have cell walls. Because of this, cytokinesis proceeds differently in plants than in animals. In plants, following division of the nucleus, a cell plate forms. We can see that up here. Notice the cell isn't being pulled inward right here. If this was an animal cell, we'd see these parts of the cell being pulled inward, the membrane being pinched. Instead, what's happening is this cell plate is forming between the membranes. The cell plate separates the 2 new nuclei, and the cell plate then develops into new membranes and cell walls. So this is the difference between cytokinesis and animal versus plant cells. It all has to do with the formation of a cell plate, which then becomes the new membranes and cell walls. Let's look at a few MCAS questions. The illustration shows a phase of mitosis. Which of the following statements describes what is occurring in this phase? A. The chromosomes are duplicating their DNA. B. The copies of each chromosome are separating. C. The chromosomes are moving toward the center of the cell. D. The homologous chromosomes are preparing for crossing over. Well, the answer, let's take a quick look at the image. Notice what's happening here. These are our centrioles, these are our spindles, and they're pulling on the sister chromatids. They're pulling them away from each other or apart. And so the answer is B. The copies of each chromosome are separating. Because the sister chromatids, remember, are just copies of each other. The diagram shows part of a cell process. Which of the following describes the expected end products of this process? Let's take a look at the image. So we have a nucleus with these are actually unreplicated chromosomes. But now we have, in the second stage, we have replicated condensed chromosomes. Looks like the nuclear envelope is dissolving. The centrioles have become clearly identifiable. We have the spindles extending from them. Here we have Well, this is obviously metaphase, right? We have the chromosomes have lined up in the middle, and the centrioles with the spindles are about to pull the sister chromatids apart. So obviously this process is mitosis. So let's take a look at our possible answers. A, two cells that are genetically identical to the parent cell will be the products of this process. B, four cells that are genetically identical to the parent cell. C, six cells, or D, 8 cells. Obviously the answer is A. Two cells that are genetically identical to the parent cell, because this is mitosis, and that's what mitosis produces. The diagram shows a plant cell at a particular stage in the cell cycle. This stage occurs immediately after which cellular process? So we have a cell plate, and that is part of our clue. Of course, we also have two nuclei. And so, of course, our answer is D, mitosis. The plant cell has already undergone division of the nucleus, and that's mitosis in the narrow sense. And a cell plate is forming, and that's going to create the new membranes and cell walls between these new cells. This video was about mitosis. Specifically, it was about the stages of mitosis. And PMAT is an acronym intended to help us remember the names of those stages, which are prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and the pairs of sister chromatids become visible. During metaphase, the chromosomes line up in the middle of the cell. During anaphase, the sister chromatids are pulled apart or away from each other. And during telophase, two new nuclei develop. Mitosis in the narrow sense is followed by cytokinesis, division of the cell's cytoplasm. Cytokinesis is when the cell divides in two. And in plant cells, cytokinesis involves the formation of a cell plate.